This pharmacology lecture was recorded in spring of 2015 on the topic indicated at the top of the slide. Please note that this lecture is quote-unquote as is, meaning that at times the audio quality may not be sufficient to completely hear everything as it was initially presented. There is no substitute for regularly attending class.
Okay, everybody, so we are going to embark on the next part of Chapter 2. And in this next part of Chapter 2, we're looking at something called signal transduction. And so signal transduction is going to refer to how a signal moves across the membrane to result in a response. Question? Yeah, so you can always log back in as long as the time's running. So it shouldn't be a problem. If the time has stopped, then you won't be able to log back in. So that's a problem. But remember, it's 2 points. So each quiz is 2 points. So don't go into panic attack mode if it doesn't work out. It's really just meant to keep you on track, right? To learn and to be excited about pharmacology. Right? So, all right. So the idea here is signal transduction, and I'm just going to set my watch so I know I'll take a break. Okay. So the idea here is, how do we go from drug binding at a receptor that we talked about last class, the receptor theory of pharmacology? How do we go from the binding to a receptor to a response, right? Because this is an experimental science that has for its purpose the study of changes brought about in biologic systems by chemically acting substances. So how do we get those changes? And so really, we have to first stop and realize that the cell is the smallest functional unit of the tissue. Right? And the cell has some interesting components that demand respect. Right? So if you're playing against the Lakers, even though they're not that good anymore, you still have Kobe Bryant on the team. You have to respect his skills. Right? You have to give respect where respect is needed. And so here we've got this membrane. And so the membrane is a barrier for many things. Right? So if you happen to be a peptide, right, there's a lot of drugs now in your pharmacies, peptide drugs. Anybody dispense like infliximab or any kind of injectables? Have you seen any? Anybody seen them? Right, peptide drugs. So these are big monsters, humongous. So they're never going to get into a cell. So their task is to get up to the cell. Just get up to it and bind, and something can happen. Right, so their task is to get up to it. So we have transmembrane receptors. And transmembrane receptors have something unique about them that's worth pondering. All right, they have a portion of their structure that goes through the membrane. We call that the transmembrane spanning domain. And some of these receptors have multiple ones, so they're called transmembrane spanning domain. We have an external portion that binds to the drug. It's called the drug binding domain. And we have an internal portion. And we sometimes call it the cytoplasmic domain. Cytoplasmic domain. So, for drugs that are unable to enter a cell, sometimes it's good enough to approach and bind on the outside. But think about that for a second, right? So again, back to our example. This classroom is the inside of the cell. And the ceiling would represent the cell membrane. And so something out there is affecting me in here. But I don't see it. But it's affecting me in here. So how does the message from the outside get in? How does the message from out there get in here? That's the process of signal transduction. Right? So the message out here, but something's happening in here. That's pretty interesting. That's like how does that happen? Right? And so this arrow is pointing to the word messages, which is pointing to the word function, cellular function. So something's happening out there that's resulting in messages in here that's affecting the cell's function. And so the question is, how is this arrow really looking in reality? Is it a single message? Is it a relay? Remember in the Olympics, one of my favorite track and field events is the 4x4 relay with the baton, right? They run with the baton, and they pass off the baton, right? And then the next person runs, and they pass off the baton, and the next person runs. It was the only time I ever won a medal in track and field, in the 4x4 relay, because I was stupid, right? But I was fast enough to make it, right? Because the lead was so humongous, the one meet that I finished today. So it was cool, but neither here nor there. Is this message multiple? Is it single? What's going on? How do we get from binding to effect? So the membrane poses a barrier for many drugs, right? So my heart's beating, right? Norepinephrine. Norepinephrine's working on a cell surface receptor. It's not working inside the cell. It's working outside the cell. So knowing how it works will be important in our course. Knowing what kind of receptor and how the receptor is wired, and so on and so forth. Now, interestingly, right, there's probably hundreds or maybe thousands of drugs that you can potentially use in a patient. And they all are going to basically work either by stimulating a transmembrane receptor or by acting at an intracellular receptor. So you could just break down that 1,000 into groups of those which work at transmembrane receptors and those that work at intracellular receptors. And so intracellular receptors have some common features. They have a drug-binding site, right? But they're found inside the cell. And so the drug that binds to them is somehow able to enter the cell. It gets inside. And it usually gets inside simply by passive diffusion, right? Simply by passive diffusion. And so thousands of possible drugs, but really two major types of receptors, transmembrane or cell surface, intracellular receptors. And so, even though there are multiple types of possible ligands, there's really only a handful of mechanisms by which they can work. And that's kind of humbling. If you think about it, hundreds of possible drugs, but really only a handful of receptor-mediated responses. And so we're going to spend some time on them today, talking about signal transduction, realizing that most receptors are proteins for reasons we've already discussed, right, for the very large diversity of amino acids and how they can be arranged as peptides. And so we have cell surface receptors, intracellular receptors, and inside this category, we have intracellular receptors, which are enzymes, or intracellular receptors that are not enzymes. So we'll spend some time talking about them today as well. Now, again, for each of these signaling pathways that we'll talk about, it's really based on getting into the cell, if the receptor is in, or being able to approach the cell if the receptor is on the surface. Right, so that lipid bilayer in the membrane of the plasma membrane is a big barrier. But there are other membranes in cells, right? Organelles are membrane-bound structures. And so some of the things we're going to talk about today end up getting into the nucleus of a cell. And the nucleus of a cell is circumvented by a membrane. Right, so some of these drug-bound receptors that go into the nucleus are going to have to get through the membrane in order to get to their target. So, as we're going to see this afternoon, we're going to talk about lipid-soluble ligands that cross the membrane and act on intracellular receptors. And then we're going to talk about transmembrane receptor proteins. And so, I guess the simple model I can draw for you is that this is a receptor, and this is the membrane. The transmembrane receptor protein looks something like this. Drug binding site. attach in the same structure to an enzyme. So the drug binds, there's a conformational change that activates the enzyme, which is part of the structure. Right? So the cytoplasmic region is an enzyme of some sort. And so what kind of enzymes might we see here in these transmembrane receptors that are enzymes? We can see something called receptor tyrosine kinases, receptor serine threonine kinases. We can see receptor guanylyl cyclase. So you see the suffix ase, kinase, cyclase, telling you that this is an enzyme. A kinase is an enzyme that phosphorylates something. Right? So a kinase will take a substrate and will phosphorylate it. Does phosphorylation do something to the substrate's kind of electrical force field, if you want to call it that? Did you just go from substrate to phosphate group with ionizable hydroxide? Did you just change the electrical nature of the substrate? I mean, what do you think? You've gone from substrate to something that has charge on it, right? So phosphorylation is a signal in cells that can be detected. It can transduce something, right? So the enzyme that does that is kinase, and it uses ATP, breaks it down to ADP, and puts the phosphate onto that substrate. Right? And so the phosphorylated substrate is different than it was before it was phosphorylated. Right? You gotta realize, like, the cells have no senses, right? They have no eyes, they can't see what's happening. But they can sense changes in electrical potential, right? They can sense changes in structure by physical change. And so substrate phosphorylation is a big deal because it can cause signaling to go. So what does phosphorylation? Kinases do phosphorylation. What kind of kinases do we see in cells? Two major types. We see we see tyrosine kinases or serine threonine kinases. Now, when we say tyrosine kinase, we need to know a little bit more because that's kind of a vague term. Are we talking about receptor tyrosine kinase, which means the enzyme is linked to a receptor? Are we talking about soluble, freely floating tyrosine kinase? Right, so if you're a soluble kinase, that means you can just swim around the pool of the cell. You don't have to reach the membrane. Right, you're swimming around, you're over here by the lysosome, right? You make a stop over here by the mitochondria. You're floating around wherever you want to go. But a receptor tyrosine kinase, where are you going to find it? I'm looking. Oh, there it is. It's at the membrane. Right, it's part of the receptor. Serine threonine kinase, same thing. Receptor serine threonine kinase needs that membrane. Receptor guanylyl cyclase, this enzyme is tethered as part of the receptor to cytoplasmic phase. And so that's different than soluble enzymes floating around wherever it finds itself. In the backstroke, wherever. So, RTK is a big catchphrase today in pharma, and I'll explain why in a few minutes. But receptor tyrosine kinase is a kinase that's going to phosphorylate a substrate on tyrosine. But the truth is, it's not really just phosphorylating the little sticky amino acid tyrosine that's floating around by itself. So where would you find tyrosine if it's not just floating around by itself? Where might you find it? And what kind of macromolecule? Protein, right? You'll see it in a protein. So imagine this wall is not made out of sheetrock, but made out of bricks. And every brick represents an amino acid. Right? So you got this polypeptide of 1,000 amino acids. And now let's say that all of the tyrosines you painted yellow. Maybe you got 8 yellow bricks in the wall. Those are the residues that can get phosphorylated by the kinase, the tyrosines and the peptides. So a tyrosine kinase phosphorylates tyrosines and peptides. If you were to look at tyrosine R group, remember what it looks like from biochem? What do I got here? What kind of group? OH, right? So OH is a perfect site for phosphorylation, right? So this is where the phosphorylation happens. Right, so tyrosines are special amino acids because they're phosphorylatable. That's the word. Right? Same thing for serine and threonine. It's just that their hydroxy is not para to the R group. Right? They're ortho or they're meta to the R group. So you have a different type of kinase called serine threonine. Right? But that's not why you see all amino acids phosphorylated. The ones that have free hydroxy groups are phosphorylated. Now, guanylyl cyclase. ACE means enzyme. Cycle means circular. So this is an enzyme that makes a circle. It's an enzyme that makes a circle. If it's a guanylyl cyclase, what's the name of the molecule? Cyclic GMP. Cyclic GMP. So guanylyl cyclase takes GTP and converts it to cyclic GMP plus GTP. Cyclic GMP is a second messenger. And for the moment, what I'd like you to think of, and I don't know if you're going to see it the way I see it, but if GTP is some kind of nucleotide that's got 3 phosphates on it, and this is a very poor drawing, so please don't tell Dr. Portafara or Dr. Talili or anyone. It's a horrible drawing. No, something like that. Right? So if we look at this molecule, right, GTP, I'm arguing that it's linear. It's a bent wire. You see it's kind of like a bent wire that you can stretch out. I call it a line. It's a Zigzag line. Right? But that line gets converted into cyclic GMP, which is a circle. It's a cyclic compound. If you don't remember, look back at that. And what is the name of this linkage called? What kind of linkage makes the cyclic compound? A phosphodiester linkage. Does that sound familiar? Phosphodiester. So this linkage, phosphodiester, phosphodiester causes the circle. So what kind of enzyme degrades this cyclic compound? What would you call it? An ACE that degrades phosphodiesterase? A phosphodiesterase. Phosphodiesterase. So sometimes we call it PDE. Phosphodiesterase. I'm going to come back to that in a moment. Right, so back to the business at hand. Drug can bind to a receptor and stimulate the activity of an enzyme, which is part of its own structure. And so the enzyme might be a tyrosine kinase, the enzyme might be a serine threonine kinase, the enzyme might be the guanylyl cyclase, but whatever the case may be, we have a biochemical effect. Something has happened. We've changed the substrate into something electrically different. We've started to accumulate a lot of second messengers. We've done something different. Right, so you're going to see these terms, receptor, protein. Oh, wait, that's not working. Protein tyrosine kinase. You'll see those other terms as well, and I'm going to talk about that type of signaling as we move forward this afternoon. In addition to receptors that are linked to enzymes, we have some receptors that are linked to channels. These are called ligand-gated. ligand-gated ion channels. And so the drug binds, there's a conformational change, the channel opens and something can enter or something can leave. And what will have an impact there is the concentration gradient. Right, so sodium will come in, potassium will leave. Right, this is kind of like when you go to the library and you scan your storm card and the text turnstile moves and lets you in. You've done that, right? Okay, so that's like the ligand-gated channel. And we'll talk a little bit about that today too. And lastly are the G protein coupled receptors. And so in these receptors, we have a transmembrane receptor, right? Drug binding region, but it's not linked. It doesn't have its own enzymatic activity. But it needs to have some kind of enzymatic activity to have a biochemical effect. And so in this case, the drug binds, and there's a coupling molecule called a G protein, and it couples binding to enzyme action. It goes in between the receptor and the enzyme, and the receptor and the enzyme. The receptor, the G protein, and the effector enzyme, because it causes an effect. Maybe the effect is accumulation of second messenger like cyclic AMP or cyclic GMP, or another second messenger like calcium. But you see there's a go-between here. There's a go-between between the drug binding and the effector. And the go-between is called the G protein. And it shuttles back and forth. Alright, so we're going to now look at some of these different pathways. The first that we'll talk about is the lipid-soluble agonist. So this one can just cross the membrane. Doesn't have a problem. And on this picture, the outside of the cell is out here, where my pointer is. Right? These would represent the polar head groups of the membrane. These would represent the hydrophobic tails of the cell membrane. This would represent the inside of the cell. And so here, lipid-soluble ligand comes in and binds to an intracellular receptor. And then something happens. The intracellular receptor may be an enzyme, or it may not be an enzyme. If it's an enzyme, then something's going to change in response to drug binding immediately. If it's not an enzyme, then something else is going to happen downstream. There may be multiple steps. Just excuse me for a moment. I'm really not leaving you. All right. Okay. So maybe an enzyme, or it might be a regulator of gene transcription. A regulator of gene transcription. So back when I was in college, I used to have a radio show, and I used to play "Regulate" by Warren G. as my background music that I was singing. It's a good song. You should listen to it one day. All right. So regulator of gene transcription. So I want to talk about that. So. All right, so I'm in the nucleus now. I'm at the center of the universe, right? I'm talking to you about the DNA. And the DNA has what within it? What do we call things on DNA? It's code for protein. Genes. Right? So we got lots of genes on DNA. Now, if we were to just look at DNA as a molecule, we can kind of break it into two regions, or two different types. We can talk about coding DNA, and we can talk about non-coding DNA. Coding DNA contains exons that make up genes when they're spliced together. Right? So the coding DNA codes for genes, and the genes end up being what? DNA gets converted into what kind of molecule? RNA. And RNA gets converted into what kind of molecule? Protein. Now, what kind of RNA are we talking about here? Messenger RNA. Very good. So there's other types, but for the purposes of regulator of gene transcription, we're talking about the transcript, the message, right? So when you go from DNA to messenger RNA, that process is called transcription. What's the process of going from message to protein called? Translation. And where does translation happen? What organelle is doing it? Ribosome in what area? So, rough ER, right? The RER. So translation is pretty much a done deal. So 99% of the time, if a transcript is made, it will be translated. Translation is not usually rate limited. Rate limited is transcription to make the message. The message takes longer to make. And so we make the message from the DNA. And particularly the genes code for the message. Now, I've drawn some boxes around genes, and I've shown you that there are definitely sections of DNA that code for protein. But what about these sections in between? What would we call those? Non-coding regions. Right?
Class.
You have in every cell of your body 9 billion bases of DNA. And if you had $1 for every base, you'd be done. Right? We have 9 billion bases of DNA in every cell of your body, and that codes for something like 30,000 genes. Right? And in any particular cell, you got something like 6,000 to 7,000 genes that are expressed all over. So there are some genes which are in all cells. They're called housekeeping genes. Things like actin, things like tubulin, right? Housekeeping genes that everybody needs. We all need basic foundation and walls and a roof. And then there are cell-specific genes. So there are certain genes that get expressed in neurons that are only expressed in neurons. And certain genes that are expressed in adipose. It's only expressed in adipose. Certain genes expressed in muscle that are only expressed in muscle. We've got cell-type specific genes. Okay, so back to it. So the process of transcription. So yes, there are coding regions, but then there are non-coding regions. Some of the non-coding regions you may have heard of are things like introns. Those are pieces of DNA that don't get made into a gene. Sometimes they're called junk DNA, but we're realizing that they're really not junk. Another type of non-coding DNA are promoters. Now, promoters are regions that flank genes, regions of DNA. And what promoters do is they stimulate transcription of the gene. They promote transcription. So let's think for a little bit before we go there. To go from a DNA molecule to an RNA molecule, what kind of enzymes do you need? That's a good guess. It ends in ACE. It's called polymerase. And what kind of molecule is that? RNA. It's called RNA polymerase. So you need RNA polymerase to ride down a gene like a train on a train track and make a RNA copy. How about if we, for a moment, pause? Let's say we want to copy our DNA and go into mitosis. So we're a little you. We're you at 8 years old. And you went through a lot of mitosis to become you at 20 or 22 or 25 or however long. Right? So you went through a lot of mitosis to go from little you to you. Right? So there was cell division and DNA replication. So what enzyme replicates DNA? DNA polymerase. Very good. So DNA polymerase happens to be a druggable target for what kind of cancer, right? Because we want to prevent DNA replication. But we're not really talking about DNA polymerase today. Today we're talking about RNA polymerase. Now, RNA polymerase forms a complex with another group of protein. I'm calling it TM. And TM stands for transcriptional machinery. There's something like 12 or 14 proteins in transcriptional machinery. They all have original names like TF2A, TF2B, TF2C, TF2D, TF2. You get the point? Transcription factor. So this is made up of a lot of proteins. This is a monster in the nucleus. It's like a space station. It's like floating around in the nucleus. And it's waiting to transcribe things. But how does it know what to transcribe? There has to be a signal to tell it to come to the gene that should be transcribed. And the signal comes from the promoter. Right, so a promoter, probably the most easy to think of promoter, is Don King. You know him? He's got the really tall white hair, and he's at every boxing match. He promotes the heavyweights, right? So he goes around and he books the fights for the boxers. Right? So he goes around to Atlantic City and to Las Vegas and to Mexico City and to wherever else the fights are. And he's like, I got this great boxer named Bilak. He's going to take on Klitschko. And he's going to knock him down. And we want that person to be $2 million because I'm going to get $400,000 for promoting the fight. And I'm going to advertise it on HBO. I'm going to advertise it on Univision. And I'm going to put it everywhere. Right? So the promoter gets the word out. So the promoter gets the word out that the gene should be expressed. But it only gets the word out when a receptor bound by drug binds to the promoter. And so where does that receptor come from? It comes from here with the drug-bound receptor that falls into the category of regulator of gene transcription. So the drug-bound receptor binds to the promoter, and here's how it goes. It signals the transcriptional machinery to come and transcribe the gene. Right, so the best example I could give you of this is when I used to live in Richmond Hill and I would take the Q24 bus to school. I was going out to Atlantic Avenue. And if I wasn't at the bus stop yet, would the bus stop? Out of the kindness of his heart, wait for me? Or would he keep going if he didn't see me at the bus stop? He would keep going, right? So in this example, the bus stop is the promoter. If there's somebody there waving their arms to the bus, hey, stop, hail the bus. Right, the promoter site is occupied, and it's signaling to the transcription machinery to come and transcribe. You'll get transcription of the gene. So this will recruit the transcriptional machinery, and you will express the message. Okay? Now, if I'm running to the bus and I'm waving my hands, but I'm not at the bus stop, is the Q24 stopping for me? No. Remember, the transcriptional machinery is like a humongous machine. It's there in the nucleus. It doesn't exactly know where to go. It has to be told where to go. Where does it go? It goes to where the promoters are asking it to come. So what's exciting about this, right, is that you have a drug-bound receptor. The receptor has a drug binding site. But it also has a portion of its structure that binds to what molecule? What is the promoter made out of? It's a non-coding piece of DNA, right? So this is cool because the receptor binds a small molecule drug, but it also binds DNA. That's called a DNA binding domain. Now, what do you think? Do you think that interaction is covalent? No. Right, that's not covalent. It couldn't be covalent because it would never turn off. Right, so it's a reversible kind of binding. So, if the drug falls off the receptor falls off the DNA, and the transcript stops coming. So that's what a regulator of gene transcription does. It does something to transcripts. Does that make sense? Do you feel like you know what a promoter is now? Okay. All right. Let's go back a moment. Before we go to the regulators of gene transcription. Here's an example of a lipid-soluble agonist coming into the cytoplasm, binding to an enzyme, and then stimulating the enzyme's activity. That's different than what we talked about a couple classes ago when we said dihydrofolate reductase can be inhibited by methotrexate, the enzyme's blocked. Here we have a similar situation. Agonist stimulates the enzyme. The enzyme gets turned on by nitric oxide, and GTP gets converted to cyclic GMP. One molecule binding the enzyme and stimulating could result in thousands of cyclic GMPs being created. Right? Because this reaction is going to keep happening until nitric oxide falls off. Okay, so the return on investment is good. Like if I was investing in signal transduction, I'd invest in the second method because I'd have a lot more than if I invested in the first method. Right? So single binding, lots of second method. So let's step back for a moment. So nitric oxide is coming into this tissue. This tissue is smooth muscle. Let's call it vascular smooth muscle. Where is the nitric oxide coming from? Do you remember which cell of the blood vessel releases it? So I'll give you a hint. The old name for nitric oxide was endothelial-derived relaxing factor. So which cell is making it? Endothelial cells, right? Endothelial cells is making it, and it's awesome. It's like a gas. It's like flowing out of the endothelial cell into the smooth muscle below. And the smooth muscle below is relaxed. The smooth muscle below is dilating. What's happening to the blood pressure? Going down. Right? So nitric oxide has a vasodilatory effect. Why? Because it increases levels of cyclic GMP, and cyclic GMP is relaxing the muscle. Okay? We good? Okay. By the way, the person who discovered that EDRF was nitric oxide was from downstate. So, right here in New York. All right, back to the other type of receptor. So, lipid-soluble ligand diffuses in, binds to an intracellular receptor. The intracellular receptor goes through some kind of conformational change and moves to the nucleus, where it's going to sit where? On gene promoters. Right? And so, this type of mechanism is used by lots of lipophilic agents. Okay? Glucocorticoids, like cortisol, do this. Mineralocorticoids, like aldosterone, does this. Sex hormones, like estrogen and testosterone, do this. Vitamin D does this. Vitamin A does this. Thyroid hormone does this. This is a very conserved type of approach. Drug coming in, diffusing through the membrane, binding to a receptor, ending up through this, affecting gene transcription. So, in evolutionary biology, many people think that long ago there was some kind of common mechanism, right? A common precursor that from which all of this comes. And so things like glucocorticoid receptor, vitamin A receptor, vitamin D receptor, they are said to belong to a superfamily, right, of intracellular receptors. Now, remember how I said the drug-bound receptor uses something like a storm card to get across the membrane? The storm card that they use is called a nuclear localization sequence. It's a special password that you have to have, a structural moiety that lets you get to the nuclear membrane and pass through. It's like a storm card, if you will. And so what we know is that, well, how come the receptor just isn't in the nucleus, even if there's no agonist then? If it's got a nuclear localization sequence. And the answer is, well, usually the nuclear localization sequence is blocked, or it's obstructed by an interacting protein. The protein blocks it, keeps this receptor in the cytoplasm. The protein that blocks it in the world of glucocorticoids is something called heat shock protein 90. 90 refers to kilodaltons, how big it is. Heat shock protein refers to how it was first identified. So back in the 50s, people were interested in understanding fever. Right? Fever in the morning. Fever all through the night. Fever. Right? Fever all through the night. So what they did was they took cells and they put it into a hot water bath that was 42 degrees Celsius to mimic fever. We think the cells were happy? Cells were not happy. But one of the things that scientists observed was that there was a series of proteins induced by heat shock that came to the rescue. So what you do when you heat a protein, you give it kinetic energy, you start to unfold it, and if you unfold it, it's no good if you're a protein. Right? If you unfold that active site, it's not an active site. Right? So these heat shock proteins are also known as chaperones. They come up to the proteins that are starting to unfold, and they squeeze them, and they hug them, and they try to keep them in their three-dimensional conformation. So heat shock protein 90 was known for that purpose, but it was later found that it actually binds to the glucocorticoid receptor, keeping it in the cytoplasm. So here's our model. Drug comes in, binds to the receptor, conformational change. It kicks off the heat shock protein. Now the drug-bound receptor goes into the nucleus. So it's the conformational change that gives HSP90 the boot. And then the drug-bound receptor can go into the nucleus. Oops. And so before we take a break, I just want to talk a little bit about this mechanism, because if you kind of look at it from a distance, it's kind of complex, and a lot of things have to happen. Right? The drug that you administer has to get to the cell, it has to diffuse across the membrane, it has to bind to the receptor, the heat shock protein has to fall off, the drug-bound receptor has to leave the cytosol and go into the nucleus. In the nucleus, it has to find its promoter site, it has to bind to the DNA, it has to hail the bus, central machinery, the RNA polymerase has to make a message has to get translated, protein has to find its way. So what do you think? Do you think that takes time? Or do you think that's very quick? It takes time. Right? So this complex pathway, is part of the reason why there's a lag period between administration of a drug and a desired outcome. Right? When you have a patient in your pharmacy that's coming in to take thyroid hormone for the first time because she's hypothyroid, she's not going to feel good the first day or two of treatment, because it's going to take time for that thyroid hormone to do all this stuff. It might even take a month before she feels good, and that's wrong, because it's a hormone. Hormones are slow on, slow off. Slow on, slow off. Right? Hormones are slow on, slow off. So we have a lag time. So this is why our favorite athlete, when they're going to play in a sporting event on Sunday in a football game, they don't go for a cortisol injection the day of the game. They go for the cortisol injection 3 days before the game, so that all this stuff can happen and that their inflammation could go down. Would this be a very effective way of learning language, for example? Would this be the way you want your neurons to kind of speak to each other? Well, I don't know, but the world would be really weird, because I would say something to you and it would take you like 2 hours to understand what I just said. And so you would say something back, and it's time to go home. Right? So it would be a much slower kind of world. Right? So this is slow on, slow off, but many drugs fall into this category. And so understanding the pathway lets us understand a little bit about time to effect, and also why when the patient stops taking the drug, they might still experience the effect of the drug for a while. So I don't know, have you had any patients on systemic glucocorticoids for a long time? Do they like how they feel? They feel kind of swollen, they feel kind of lousy, so they stop taking it, and then they still feel swollen and lousy for a while until the drug gets out. Okay, everybody. So we're going to jump into the next topic. And the next topic is the receptor tyrosine kinase. Now, the receptor tyrosine kinase, we've already kind of talked about. It's got a drug binding domain, it's got a transmembrane binding domain, it's got a cytoplasmic enzyme domain. And this enzyme domain is a tyrosine kinase. Now, this is the type of receptor that is used by things like insulin in your body. Of course, insulin, we know, is a critical hormone. If your insulin levels are deficient, you have pathology. If your insulin doesn't work correctly because you become resistant to it, type 2 diabetes, you get pathology. Excuse me. So insulin uses this kind of receptor, as do things like epidermal growth factor, or EGF. Now, EGF might kind of seem boring at first glance. First glance, epidermis, big whoop, right? Keratin, cells becoming keratinized, right? Going from the stratum germinativum all the way up to the stratum corneum, right? Getting keratinized, epidermal growth factors involved in that process. But as you can see, two of the words in the name are growth factor. And growth factors have an uncanny ability to help cells grow better than they otherwise would. So during growth, when you're little you and you're growing up to be big you, growth factors are fine. But once you've matured, growth factors act as proliferation signals to cells that really shouldn't proliferate, right? So if you have a cell that's become a cancer cell that's escaped immune surveillance, well, now it's getting growth factor stimulation all the time, it's going to grow. And so one of the growth factors that's out of balance in certain types of breast cancer is epidermal growth factor. Right? So there are certain mutations in certain breast cancers where this signaling pathway is always on. Cells are always getting grow signal, grow signal, grow, and they do grow. And so there have been drugs designed against this particular growth factor, antibody drugs. The drug I'm thinking of right now is called Herceptin. Have you ever seen it? If you haven't, that's okay, but it works by neutralizing EGF signal. Platelet-derived growth factor, another signal to grow. So these types of ligands use this type of receptor to get cells to grow better. And so before I kind of move on, I would like to point out that insulin itself is a growth factor. Right? So if you've ever seen, I don't know if you've had the pathology lecture with Dr. Resnick yet. Right? She shows you pictures of the children that used to not have insulin, right? Type 1 diabetics, how they'd be kind of not have much body mass. Right? They'd be very lean. They wouldn't have much muscle. They wouldn't have much anything, and they wouldn't live very long. Right? And so insulin helps your body grow during development. It works with growth hormone. Now, the problem with insulin is that insulin helps tumor cells grow good too. So if I take tumor cells in my lab that are growing on a culture dish, and I give them insulin, they grow better than they otherwise would. They start to grow off the plate. I have to seed them more frequently. Right? And that's true of many cell lines that I've worked with in my lab. Right? So I've seen with my own eyes that insulin helps tumor cells grow better than otherwise would. So I don't mean to have a huge drugs and disease talk here, but one of the things we know is that obesity often co-presents with something called insulin resistance. So many type 2 diabetics, they have insulin, but the body doesn't use it right. Right? The target tissues of skeletal muscle, of liver, and of fat kind of don't respond to it correctly. So their pancreas is spilling out a lot of insulin, but the target tissues are resistant to insulin. Have you ever heard of this term, insulin resistance? Right? So insulin resistance and obesity kind of go together. And so there's a theory that's kind of starting to be put together out there in the literature that suggests that hyperinsulinemia, having too much insulin like an insulin-resistant person does, isn't really fixing the diabetes problem and may be increasing risk of cancer because insulin may be having this growth effect. Right? So It's starting to be pieced together. You know, time will tell. When I get you in fifth year in drugs and disease, I might know more about this hypothesis. But clearly, insulin is a growth factor. And so, you know, that's a good thing in normal growth and development. It may not be a good thing in certain disease states. And so this particular slide I'm going to quickly go past because I'm going to tell you all of this stuff now in the next. So how do receptor tyrosine kinases kind of transduce the signal from the out to the in, right? Epidermal growth factor is a big protein. It doesn't get into cells. It gets up to the membrane, but it doesn't get into cells. Insulin is a big protein. It doesn't get into cells. It gets up to the membrane and works on a cell surface receptor. So how do these things work? Well, in a simple model like I've drawn for you here, those blue circles would represent insulin. And so they're going to bind to their receptor, and the receptor is going to exhibit a conformational change, which I've artistically drawn for you as this little flap coming out. Right? It doesn't really necessarily look like that, but there's a conformational change that occurs that results in a pairing up of receptors. Okay, so the drug binds, the receptor exhibits a conformational change, and it partners up with a nearby receptor. That process is called receptor dimerization, forming a dimer, two parts. In pharmacology, as you progress through the curriculum, you'll learn about receptors that can trimerize. But here, today, the theme really is dimerization. And so the receptor dimerizes, and I've pointed out for you these Ys. That Y is the single letter abbreviation for tyrosine. Right? Why not T? What is T the single letter abbreviation for? Which amino acid? Threonine. Very good, right? So Y is the single letter abbreviation for tyrosine. These tyrosines, because this is a receptor tyrosine kinase, are going to phosphorylate. Going to be phosphorylated by the catalytic domain of the receptor. And so sometimes we call this autophosphorylation, phosphorylation of self. Right, so the drug binds, there's a conformational change, there's a dimerization, there's a coming together of the catalytic subunits and the phosphorylation of the tyrosines. At this step, the receptor becomes a kinase and starts to phosphorylate other substrates in the cytoplasm that come up to the membrane. Right, and that looks something like this. Right, so the substrate can come up to this kinase and come out phosphorylated. It's different than it was before. It's got some oxygens, some ionized oxygen groups. It's electrically different than it was before, the phosphorylated substrate. Now that phosphorylated substrate is going to have an impact, or going to relay the signal from out there. And so what is the phosphorylated substrate going to do? Well, it depends. So phosphorylation of substrates in various tissues may have different effects. So I'll just use one example in our discussion since we're talking about insulin. But we can see lots of things. We could see altered membrane transport of protons or other ions. We can see altered membrane transport of certain metabolites. Okay, we can sometimes see phosphorylated substrates going into the nucleus and affecting gene transcription. In the case of insulin receptor, the name of this particular substrate is called IRS, insulin receptor substrate. And that insulin receptor substrate, when it gets phosphorylated, causes increased expression of a glucose transporter on the surface of target cells like liver cells, fat cells, skeletal muscle cells, so that all that sugar can leave the blood and enter target tissues. Okay, so insulin, for example, is going to cause increased uptake of glucose and amino acids. It's going to cause formation of the glucose storage form called glycogen. It's going to cause formation of triglycerides in the cell. So one way to think about insulin is as a anabolic hormone. It causes building. Right? Catabolic catastrophe, right? Breaking down things. Anabolic building up. Insulin has anabolic effect. It causes target cells to take in sugar and build it into glycogen. It causes target cells to take in amino acids and build them into proteins as a storage form. It causes target cells like fat cells to store fatty acids as triglycerides and stored energy for a time when there is no glucose because of fasting or prolonged activity where you can't eat. Okay, so phosphorylation of the substrate can do one of many things. It's going to be tissue specific, and it's going to be agonist specific. It may even alter gene expression. So you might say, wow, that's a lot of steps, and is it really necessary to know all these steps? And so the answer is, well, I think it is. RTKs represent a very hot area of drug discovery research right now in pharma. In fact, there's been 2 new drugs recently approved that target receptor tyrosine kinase. A drug called imatinib and a drug called lapatinib. Right? And if any of you have ever had a course with Dr. Chen, he studies those drugs in his lab. And so why? Well, we talked about growth and proliferation. If you can inhibit receptor tyrosine kinases like EGF that's overactive in breast cancer, you can reduce cell growth and cell proliferation. Right, so pharma is targeting all aspects of the pathway. They're looking for antagonists that prevent dimerization. They're looking for antagonists that will block autophosphorylation or substrate phosphorylation, and so on. In fact, those 2 drugs I mentioned to you, they block substrate phosphorylation. So in your 40-year career, right, so picture it, it's now 2055. Right? It's 40 years from now. You're getting close to the end of your career. Right? Hopefully I'm still around. I'm hoping. But most likely you will see more drugs that affect tyrosine kinases as you move forward in your curriculum and in your career. Right? So it's 2055, and some new drug comes out that affects this pathway and inhibits cell proliferation and is useful against certain tumor, for example. Now, I'm going to, well, no, I won't completely skip this, so, because I'm going to talk about the words in the next slide. But now I want to talk to you about a phenomenon observed with receptor tyrosine kinases that's kind of unique. And here we'll talk about the EGF receptor, but our discussion isn't exclusive to the EGF receptor. And so the concept I'm trying to get across here is the concept of receptor downregulation, which when you use the word downregulation, you mean fewer receptors present in the tissue than there were before. You actually have fewer receptors in the tissue than there were before. Because of what? Because of this phenomenon called receptor downregulation. And what is that? That is when the rate of endocytosis and degradation of a drug-bound receptor exceeds the rate of new receptor synthesis. And so let me try to show this to you in the pictures that are coming ahead. So I'm going to pass through this slide and the next because this is probably more detail than you need right now. And hopefully you've heard about things like, you know, the clathrin-coated pit, the coated vesicle, the uncoding, and this idea of endocytosis, receptor-mediated endocytosis. Now, this is how we think some of those nanoparticles get into cells, if you've ever studied nanoparticles as vehicles for drug delivery. But again, I'm going to go to the next slide, which I made as a simpler version, just by hand. And so here we are. We're dealing with receptor tyrosine kinase. Our example will be EGF receptor. And way at the top, and I apologize I don't have my pointer, way at the top we have a tissue where all the receptors are bound by agonists. And so in a continued presence of stimulation, receptor tyrosine kinases pinch off with the vesicle. In the continued presence of stimulation, receptor tyrosine kinases pinch off into vesicles. So here's that process happening. So we're starting to bud off a vesicle, we're starting to bud off more of the vesicle, and eventually we've internalized these receptors into a vesicle. And what about the surface of the tissue? Does it have as many receptors as it had before? No, right? So the surface of the tissue is naked now. There aren't as many receptors as there were before. So if an agonist were to come up to the tissue, would it cause a response? No, the tissue would be not sensitive to the agonist. Right? So if you're looking into this situation through some kind of experimental method, you're looking at the tissue and you see all the receptors are bound. I don't understand. Why has the tissue stopped responding? I don't get it. Right? But experimentally, we see with receptor tyrosine kinases that under prolonged stimulation, they'll pinch off and enter the cell into a vesicle. And then there will be a period of time where there are less receptors on the surface. We would say that that's downregulation. There's not as many receptors there as there were before. But the fate of this pinched off vesicle can be one of two things. On the left hand of this model, the vesicle fuses with the lysosome, the organelle that cleaves and destroys. So if the vesicle fuses with the lysosome, what happens to the receptors? They get degraded. Right? They get destroyed. So if their receptors are being degraded, what do you think has to happen to make new ones? We have to have new transcription, right? We need to have the genes for those receptors get transcribed, translated. We need to have those proteins get inserted back into the membrane. Do you think that's rapid, or is that long in the molecular time? Long, right? So if the vesicle that contains these drug-bound receptors fuses with the lysosome, it's going to take some time before we make new receptors to put back into the membrane. That's called receptor downregulation. There's an extended time interval where the tissue stops responding to the drug because there are fewer receptors on the surface than there were before. Now that's different from what's on the right side of this figure. On the right side of this figure, the vesicle is internalized, but the drug falls off the receptor, and the receptor is reinserted quickly into the membrane. So the right portion of this figure could be labeled as receptor recycling. Right? You don't have to make new receptors. You can just stick them back into the surface of the cell. So how about this in terms of time, molecular time? Is this a quick response, you think, or a long response like downregulation? Quicker, right? So yes, there are fewer receptors than there were because of this endocytosis of receptors, but the tissue can quickly reset. That situation is called receptor desensitization. Receptor desensitization. The tissue has been desensitized, but receptor desensitization is temporally short. And so a little bit of time goes by and the situation resets itself. Receptor downregulation, on the other hand, takes some time to overcome because you need to make new receptors. And so if I go back to the prior slide, which I won't do because it'll mess up my recording, but if you look at the prior slide, you'll see if the rate of endocytosis and degradation exceeds the rate of de novo synthesis, you have receptor. This one. If the rate of endocytosis and degradation exceeds the rate of new synthesis, you have downregulation. But if you have just a small interval of time that goes by and the tissue responds again, you have recycling and desensitization. So do you feel good with the difference between desensitization and downregulation? Yes, I'm recording this because I might try to put this on YouTube, but no promises. But some students seem to like it. I couldn't listen to myself again. But some people walk around with their iPod on and they listen to my lecture. Which, I mean, that's cool. Okay, so the next 3 slides I'm skipping, and I'm going to ask you to read. And they deal with something called the receptor serine threonine kinase. And again, everything you need to know is on these 3 slides. You don't have to go beyond them. Okay, so the picture, the words, to use the example of receptor tyrosine kinase to understand the receptor serine threonine kinase. Because in a lot of ways they're similar. Third example I want to talk about is the receptor guanylyl cyclase. So here we have a guanylyl cyclase which is tethered to the receptor. Is this a soluble enzyme? Or is this kind of stuck at the membrane? Right? So we call this a receptor guanylyl cyclase. And so what do guanylyl cyclases do? Cyclase, they make circles. Out of what? Out of GTP. So GTP turns into a cyclic GNP that has a phosphodiester bond. Now what cleaves cyclic GNP back to a linear molecule? What kind of enzyme? A phosphodiesterase. Very good. So, okay, back to the story. So this is an example of a receptor guanylyl cyclase, the receptor for atrial natriuretic factor. And again, factor and peptide can kind of be used interchangeably most of the time these days. But before we knew this was a peptide, we just called it factor. All right? So atrial natriuretic factor has a receptor called the ANF receptor. And so natriuretic means what? Urinating out of what? What is natri? Natremia. What is natri? Sodium. The peeing out of sodium. So when a patient urinates out sodium, what else goes out with the sodium? Water. So if the water comes out with the sodium, what happens to the blood volume? It goes down a little bit. So what happens to the pressure against the blood vessel walls? Decrease. Yeah, so decrease a little bit. Right? And so what happens to the blood pressure? Goes down. Right? So natriuretic factor is good for blood pressure. It acts in the kidney. It causes you to pee out salt and with it water. But natriuretic factor also has action on vascular smooth muscle. And so vascular smooth muscle has ANF receptors that get stimulated. A lot of cyclic GMP gets made. And what does cyclic GMP do to smooth muscle? It relaxes it. Right? So cyclic GMP relaxes vascular smooth muscle, causing vasodilation. Right? What other compound did we talk about today that activated guanylyl cyclase? Nitric oxide. Right? So this is kind of like nitric oxide, but it's not. It's ANF. And what's interesting about this is, where does this factor come from? Which part of the heart? The atria. Right? So the atria are feeling a lot of pressure against their walls, so they release this thing, which causes a little bit of relief of pressure. Right? So that's cool. And that's one of several, complex mechanisms between the heart and blood pressure and the brain and all of that. Okay, so again, ANF binds to the receptor dimerizes the guanylyl cyclases get active and start to make cyclic GMP.
That leads us to the next class of receptors, cytokine receptors. The word cytokine is actually made up of 2 smaller parts. Kine, which is part of the Greek word kinesis, which refers to movement, and cyto, which refers to the cell. So a cytokine is a protein that is released from a cell and acts on another cell to stimulate cell movement. At least that was the original definition. Now we know that many cytokines activate immune cells, cells of the bone marrow, and so on. And so what is it about cytokines? Well, these are low molecular weight, pharmacologically active proteins, and they're secreted from a cell to act in the local environment. So either to act on the cell that released it, that would be called autocrine, or to act on nearby cells, that would be called paracrine. Okay, so cytokines have autocrine or paracrine effects, and because of that, we don't really consider them hormones, right? Hormones would be synthesized in a certain tissue, released, would travel through the circulatory system to a target at some distant location. Cytokines act locally. And so there are many types of cytokine receptors. One of the receptors for growth hormone, erythropoietin receptors, interferon receptors, many other receptors that can affect cell growth and regulation, that can affect immune response, would fall into this category. And now we're going to discuss a little bit more about, like other receptors that we've seen, the cytokine receptor, when it binds to agonist, will exhibit a conformational change and will dimerize. But I'd like to point out at the bottom of this slide, you see a cellular kinase there called JAK kinase, Janus kinase. And its job is going to be to phosphorylate the cytoplasmic domain of the cytokine receptor, and then phosphorylate something else. Okay, so if we look on the next slide, we're going to see the drug binding, the conformational change. Right, the cytokine binding, the conformational change of the receptor, the dimerization, the coming together of the cytoplasmic domains. And so when that occurs, Janus kinase phosphorylates the cytoplasmic domains of the receptor, and those phosphorylated domains become a platform that attracts another type of protein to the receptor called STATs. STATs stands for signal transducer and activator of transcription. So the STATs now come up to the cytoplasmic face of the receptor, attracted by the phosphotyrosine residues. And as you notice, the STATs themselves have tyrosine residues, which will now be the second phosphorylation by JAK. So JAK will now phosphorylate a second substrate. That's why it's called Janus kinase, as a way of thinking of the fact that this has two substrates, just like the Roman god Janus had two faces. Right? So now the STATs get phosphorylated and dimerize. And that dimer moves into the nucleus where it can affect gene transcription. Now there's something like 4 different types of Janus kinases. There's something like 6 or 7 different STATs. Okay, so we have a diversity here in the genome of these types of signaling molecules which represent the JAK-STAT pathway. Again, they are going to execute signaling induced by cytokines. And a representative type of cytokine would be interleukin, for example, like interleukin 1 could do this kind of signaling to get immune cells to do something. And so this multi-step process is an interesting target for drug discovery, particularly in conditions where the immune system has been too active for too long. So for example, in a rheumatoid arthritis patient, or a patient that's got an autoimmune disease like multiple sclerosis or something, the idea might be to try to block this signaling pathway, this JAK-STAT pathway, to reduce inflammation. So we can target various places in the pathway: the cytokine binding site, the first phosphorylation, the second phosphorylation, nuclear translocation. These would all be places that could be sites of novel drug development if we were to discover a drug that could slow down or inhibit the JAK-STAT pathway without being otherwise toxic to the rest of the body, which is always a concern for any drug that's being studied. This information presented in this course is based on reference materials that are believed reliable and consistent with standards accepted at the time of presentation. Possibility of human error and ongoing research and development in medical sciences does not allow assurance that the information contained herein is in every respect accurate or complete. The information presented during the course is intended for teaching of undergraduate and graduate students in biomedical education and is intended only for teaching. Students who successfully complete this course should check the product information sheet included in the package of any drug they plan to administer or dispense to be certain that the information presented in this course is accurate and that changes have not been made in the recommended dose or in the contraindications for administration. July 9th, 2014.